Okay, let's go ahead and get started. Talk about what we've got going on. Um, so we have quiz on Friday. We have a lab report due tonight.
Take a day, relax, don't grade anything, give us another day.
I'm busy grading anyway, and I won't be able to get to it. You could get it in tomorrow and it would be fine because it's gonna be at least 3 or 4 more days before I'm done with the plasmid mapping lab. The only problem there is I have yet to remember how to extend deadlines on that. So if you take longer, you can go ahead and send me your report, but then have the patience that when I send an email to the whole class and say, okay, I've extended the deadlines, I want you to submit it, that you submit it, okay? You don't need to send it to me either. You could just remind me that I need to extend the deadlines on those. So I need to go talk to another faculty member. I don't understand what happened to it. It used to be easy to make no deadlines on those, and maybe it still is, and I'm just missing it. But yeah, so you can have longer than that if you want. Let's see. Thursday help session at
8:30?
8:30. Thank you. That's my Gen Chem one. 8:30 p.m. on Google Meet. Okay, and then I'm available Monday, Wednesdays, but appointments stay between 1 and 4. I have a student coming at 1. I have a student coming at 2. So, the 30s, probably 1:30, 2:30, if you're thinking about it for today, 3:30, something like that. Come tell me after class. Questions on stuff we've got going? Okay, your next lab report, it's going to be 2 weeks. If you've looked at the lab syllabus, there's a lab syllabus now. The next lab report will be in 2 weeks, not next week, but the week following that. So we're going to go mostly every 2 weeks. There's one where it's going to be 1 week for the rest of the semester. All right, questions? All right. All right, then what we're going to do is we're going to go on and we're going to talk about secondary structures today. We're going to talk a lot about alpha helices. Hopefully we'll have time to get to beta sheets. And one thing we want to think about when we think about secondary structures is that there are certain things we consider in terms of what shape protein make, right? The shape that a protein makes is largely determined by what shape that backbone weaves around. All right, so you have side chains as well that can flop around a little bit, but that global shape is really about what are the angles in the backbone. And so if we look at a peptide bond, let's imagine we have a peptide bond here, and we can have an amino acid over here. Let's say it's alanine. Do you remember anything about these, about amides and what happens with these from organic?
In what context? Nothing changes.
Well, right. First off, never protonate it. Never do anything. Don't change it. Okay? But we have a resonance structure here where what you can do is you can take these electrons and pop them up here, these electrons and pop them down there, and what you'll get is a resonance structure that looks like this. Right, 4 bonds to nitrogen now and 1 bond to oxygen, and so I do that. Okay, so what that means is this bond right here has double bond character, right? Resonance structures mean that I have what my real peptide bond looks like is some sort of mix between the two of these, and really this isn't 1 bond, it's closer to like 1.5 bonds. All right, so when you have a double bond, what do you know about it in terms of rotation from from organic chemistry? It's locked, right? It's not going to rotate at room temperature, which is and any kind of reasonable biological temperature. I don't know what happens in thermal vents, but you're not going to rotate these bonds very much, okay? And so, or at all. So what that means is if we look at our peptide backbone, we can look at our peptide backbone down here. Those peptide bonds are here, right? You can imagine this. Where's another one? I'm confusing myself. Here's another one. Here's another one. Here's another one. Okay? So we have these peptide bonds here, and you get no rotation. If you think about a peptide sequence, that for the backbone, we have like this atom and this atom and this atom and this atom and that atom and that atom are all in the same plane, always, right? You can think about it as a bunch of planes that can kind of rotate relative to one another. And where they rotate relative to one another is if we look at an amino acid. So this is an amino acid from here to here, right? There's the amine, there's the what used to be the carboxylic acid, that used to be the amine too. And we have an angle we call phi, and we have an angle we call psi. And so everything about that backbone is determined by the rotations of these, or the angles on these two bonds. Okay, so we have these phi and psi angles. And so when thinking about the three-dimensional structure of a protein, and so in three-dimensional structure is going to be our secondary structure, it's going to be our tertiary structure, and it's going to be our quaternary structure. Each amino acid has only two bonds that can rotate. All right, and so this becomes very important if you're thinking about, how am I going to solve that crystal structure? So remember when we do X-ray crystallography, we get an electron density map, and we try to fit our peptide into that, right? We don't really try it. A computer's going to be trying a bunch of different things and see what fits, how the atoms fit best to match that electron density map. But we only need to worry about rotating two bonds to do that for each amino acid. And certain values of phi and psi are not allowed because you're going to run atoms into each other. All right, so certain values of phi and psi are not allowed because atom collisions will occur. Right, so this becomes very, very important when you're trying to solve a structure of a protein, or if you're doing calculations of things, trying to solve that NMR structure, right? You've got particular restraints you put in that NMR structure, but when your computer program is trying to solve it, it's going to go, well, it's not going to ever try certain phi and psi angles because it just knows that they're not allowed. Does that make sense? Okay. Now I want to remind you, not showing the next picture yet. I'll remind you that I talked about how we have really three major secondary structures. And those are alpha helices. You could use an alpha. I'm just going to not because then I have to go and change the font and all that sort of stuff. So alpha helices, beta sheets, and turns. And there are a number of different names for different types of turns, but they all turn. And so you could imagine that if we have only three major secondary structures, that there would be maybe for one secondary structure, you would have a set of alpha, phi, and psi angles that are favored for that. And that's the case. And for another secondary structure, say beta sheets, we'd have a set of phi and psi angles that are favored for that. And then for terns, the same sort of thing. And so what you can do is these are angles. You can do a plot that's called a Ramachandran plot, named after the scientist who first came up with it. Okay, and what we've got over here is it's a plot of all the phi and psi angles for each amino acid in I'm going to guess this is a single protein. And if you made me guess which protein, I would say it's probably hemoglobin, because the book that I took this from loves hemoglobin, as do I. Super cool protein and super well studied because it's hemoglobin. All right, but so what this means, if we look at this plot, you see how we have clusters of things? All right, well, it turns out that this section here, this dark blue, so where we have dark blue, what it's doing is it's showing where things are more dense. This is going to be beta sheets. So beta sheet amino acids. This is going to be right-handed alpha helices. Okay? And this is going to be like left-handed alpha helices, and there are other things that might show up. But really, beta sheets, alpha helices, yes.
Is it because of where they are on the plot that you know that?
It's that I know where they are?
Like beta pleated sheets are up because they're at those certain values versus, okay.
These are the values that favor that. So we may get to beta sheets today or not, but beta sheets are an extended form of the peptide, and you can see that our angles are near 180 and 180, 180 and 180, right? Okay, so you think about this, 360 degrees you could put your angles on, 360 degrees you could put your angle plots on. Okay, and so that's why you end up with these sorts of things. And so now if I'm trying to solve a structure, now I've got even more clues. My program is probably going to start trying to fit things in here, and then if that doesn't work, then branch out from there. Does that make sense? Okay, so that's how this is useful. So what's going on here? How might I test you on this, right? I actually used this in a publication I did once. Somebody was saying they wanted me to discuss something going on with glycine residues. And, it sounded like maybe I had to go back and do more lab work. I was trying to figure out how I was going to get past this reviewer. And Dr. Sarisky saved me, as she often does. And she said, why don't you just plot it on the Ramachandran plot and see where they are? And then you can just respond with that. Okay? And it turned out I plotted them, and they were somewhere out here. And I don't even remember what the question was, but I used the Ramachandran plot. I said, oh, okay, so they're weird glycine structures, as glycine often does. We'll talk a little bit about that. And so I don't need to do any more work. And I didn't have to do any more work. Okay, but I mean, so this is a really useful thing. I've used it multiple times. And so I could ask you to plot something on a random. I want you to understand what it is. Okay, and so in terms of a question, I might give you a couple amino acids with angles and ask you to plot them on the Ramachandran plot. And so if I had, let's say I had let me show you how I would do that. If I had an amino acid here, let's say it is isoleucine, and what I did on that is, so I've got, actually we're not, so it's in a peptide. Make sure to draw my hydrogen here. And then we've got this end right there. And so our angles that we can rotate are these two. This would be my phi. This would be my psi. I could say that my psi is equal to, let's say, 60 degrees. Okay, and my phi is equal to, let's say, negative 120 degrees. I just made that up. Look at the plot real quick, or draw one on your paper if you want. Plot that. You plot one data point for that. All right, so hopefully what you did is you looked on this bottom axis, right? My phi is 60, so I'm going to come out to about 60. That's about here, right? I don't know if you can see the little spot here. And then I would go up to 100 and negative 120, and so it would be right about there. Should have, when you were thinking about it, plotted something right about there. And so if we had an angle that looked like that, we might be, something weird might be going on there. We might be concerned about it, right? If we had plotted it and it had landed here, we would have said, oh, it's probably part of an alpha helix. That makes sense how this gets used? Yeah.
So is this like a pretty universal plot? Like you can use it for whatever different proteins or different sequences, and like now if it's in this area, it'll be like an alpha helix.
Yes.
Okay.
Yes. So it's really quite universally used. Everybody's using it to look at things.
Do we need to know the values of
No.
each other?
But you need to understand it, maybe plot something on it. Maybe if I gave you the values, be able to use the plotting and do something with it. Okay, so let's go on and talk about alpha helices, right? So alpha helices, what they are these twisty things. Okay, you've seen these ribbons, right? These curly ribbons in the protein diagrams I've shown you, and I'm gonna dare to show you something in a bit. I want to go as far as I can here in case the video cuts out as a result. But essentially, they are structures. I already told you what hand helices they are, right? Can you confirm that I was correct? Yeah, they're right-handed helices, right? We look at it going around this pole. It's going right-handed in that direction. So, alpha helices. Okay, they're predominantly right-handed helices. As you saw on the plot, for at least hemoglobin, you could occasionally get a little bit of a left-handed helix, but really we're going to just be all right-handed helices in here. They have, if you look here, it says right here that from one end to another, so if we think about going around this, if we start here and we go 3.6 residues, we end up at the same point. You see how this and this are kind of the same? It's about there. Okay, so one turn is about 3.6 residues. When I say residues, I mean amino acids. So follow, you see we've got this little thing that, I don't care about the angstroms here. That's not going to be useful to us. We follow this over here. We then go around the helix. We follow this over and we end up at about the same spot. So overall, not perfect for this picture. Actually, it is really kind of, I'm on the carbonyl, I'm on the carbonyl. I think we'll go a little bit. And what that means is that when we think about the side chains, that if you do one full turn, right, you imagine I'm here, I've got a side chain hanging off. In terms of these atoms here, these purple atoms are the side chains. These purple atoms, they're not, it's a purple blob. Side chains would be a bunch of atoms coming off. Right? And so what that means is that if I go about 3 or 4 residues around, what I get is I get things on the same side of the helix in terms of those side chains. And so this, even though this is not close to this in primary sequence, they can interact. All right, so we say 3 or 4. So 1, 2, 3, 4. So I could have an interaction here, I could have an interaction there. Just gonna look at the other map. I did lose one in the back. All right, does that make sense? So we can get those interactions, and those interactions could be important. Those interactions can be important for stability. So if this was a positive charge and this was a negative charge, they'd attract each other, which would help this compressed alpha helical structure stay that way, stay in its shape. They could be, if that's a negative, it's going to push it out, going to make it not want to be an alpha helix. Okay. If I put a positive and a negative here, or I put all hydrogen bonders here, 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 and here, then that would be good for hydrogen bonding with another protein. Or these could be all hydrophobic side chains, and it would be good for packing with another protein if the other protein had the complementary polarity. All right, so amino acids 3 or 4 apart are on the same side of the helix. All right, then I want to talk about a couple amino acids. So proline, what do you remember about proline? Yeah, it's got a ring that attaches with the backbone. Here, I've got a picture of proline down here. Here's proline, right? So we make a peptide bond there, we make a peptide bond to here. So what this does is this forces a turn, really. We're forcing the sequence is going this way and then it goes that way. And it's pretty rigid because the ring is not allowing things to turn like they would. So if we think about our phi and psi angles, this is our phi angle here, and this is our psi angle here. And our phi angle, yeah, we could rotate around that just fine. But our psi angle, that's going to be restricted. You don't need to. The phi is between, so that's where our side chain comes off, right? Phi is after the amine, psi is between there and there. Okay, and I haven't really emphasized it as much with peptides as I did with the nucleic acids, but we have a direction to proteins as well. We have an amino to a carboxylic acid direction. Let's go ahead. We'll go all the way up here. Okay, and what we're going to do is say this. Well, actually, it's actually emphasized here. Amino end and our carboxy end. So there's a direction to these polymers. Okay, so proline is very rigid. And is not often found in alpha helices because it doesn't allow the alpha helix to have the angles that need to form that spiral shape. Let's say, and isn't found very often. Okay, glycine. What do you think about glycine? If anybody remember what glycine is? It's just H, right? Nothing. So glycine, what do you think? If proline's too rigid, what do you think about glycine? Glycine is super flexible and also not found. Too flexible. Okay, and then finally, actually the thing I should have said first is, what do you see holding this thing in its shape here? demonstrated over here. Hydrogen bonds. So if hydrogen bonds are going to help hold the alpha helix in its shape, and are the primary force holding the alpha helix in its shape. And that's going to be the case for all of our secondary structures. Beta sheets, it's going to be hydrogen bonds. Turns, it's going to be hydrogen bonds. Okay, it's always hydrogen bonds, and then you might get some help from some other things. Yes?
Things like isn't that also tryptophan? Is that not on something?
Tryptophan is fine. And let me tell you why. So when you look at the three-dimensional structure of the of an alpha helix, what happens is the or the side chains are on the outside. Okay, so if we think about tryptophan, which is really big, tryptophan would point out here, so it'd be good, it'd be fine. It depends on what you're packing into in the protein over here, but just from an alpha helix on its own, it's fine. So in 3 dimensions, an alpha helix, the side chains in an alpha helix point out. And so you can see that here with both this sequence, right? We've got a couple different ways. Here we have a space filling model, and you can't really see the inside as well, right? It's really tightly packed. And here we have a not so space filling model, right? With ball and stick, but so you can see down it, but there's really not space there. Okay, does that make sense? And you can see how they stick out. All right, so here I'm hoping that we don't lose our video. Haven't yet. All right, and so this is a model of, this is the PDB code for this one. I don't remember what the actual protein is, but it's 2F1G, and it's one of the ones that I'm working with in my research. A lot of things I'm working with in that. And I just wanted to grab alpha helix and look at it. I spent way too much with this one because alphabetically it comes first in my list. But here, I'm trying to, there we go. Just want to quick grab, okay. Where is that? There we go. All right, and it looks like it's near the end, so this might be easy. Nope, it's this way. Oh, come on. I must not have, okay, I didn't click the alpha helix. Let's click this one and unclick this one. Okay, so we can look at this one all by itself. And what I can do is I can show as spheres, and it looks like we are showing everything. So side chains and all. So here's my alpha helix. You see when we look down that, there's no hole in the middle. And then you can look at it like this. Can you see that the side chains here are sticking out? Let me see if I can hide our side chains real quick. Hide side chain, yes. Okay, and there's our alpha helix without the side chains. Okay, so that's what that stuff looks like in 3 dimensions. Questions on alpha helices? Okay, we're not done talking about them. But, so alpha helices, so while alpha helices are held together by this hydrogen bonding, they can be made more stable or less stable depending on the order of the amino acids. And so while they're held together by hydrogen bonding, I don't want you to forget that that's our main force. All right, so we've got a nice little alpha wheel helical diagram down here. We're not going to use it just yet, though. I want to take a simple case first. Let's say we have an alpha helix that's made from a sequence that goes E, 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 E.
Really like glutamic acid.
Right, glutamic acid. Okay. So this is glutamic acid or glutamate, depending on which protonation state we're in. I'm going to put my carboxylic acid. Okay, I'm going to put the impossible form where it's neutral, right? Remember that doesn't exist? Because we're about to talk about pH, I don't want to bias it. All right, remember that when we think about the side chain, so I've got, this sequence is going to look like this if we look at one amino acid. In its protonated state. All right. And what's the pK of that side chain, roughly? If I don't tell you, what is it? It's about 4. 2, 3, 4, sure. All right. So, do you think this alpha helix will be stable at pH, let's say 14? All right. What do you think? What's going to happen to this functional group at pH 14? It's going to deprotonate, right? And so what we're going to end up with, so we'll do pH 14 in red. You're going to end up with negative, 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 etc. Right? So I'm going to have a whole bunch of negative charges next to each other. And alpha helix is this condensed curled shape, right? Like a spring. And what it's going to want to do is stretch out to get those negative charges as far away from each other as possible. So at pH 14, this thing is going to all be negative, and it's going to stretch out. Okay, it won't be an alpha helix. Let's go ahead at green. How about pH 7? 7's a great pH, right? What about pH 7 for this sequence? What was that? It's better, but what do you think? Still, what's our biochem rule? If charges are really important, yes. Don't forget that. But if we're 2 away from the pKa, we're all in one form, right? And 7, if that's 4, take the top of our kind of range for, if 7 is 2 away, it's higher, right? So protons come off. So at 7, it's still going to be negative, 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 etc. And those negatives are going to push against each other, and they're going to stretch our alpha helix out. So over here, 14, nope, nope. Okay, and then what about if we do, I need another color, we'll do blue, I guess. What about pH 0? Not stable at pH 0? What was that? So it's 4, we're 2 away, yeah. So which form is predominant? Which of the forms? So over here, do you guys understand why they're negative?
Yeah.
So what happens at pH 0? They all become neutral, right? So I no longer have it pushing everything away. Does that make sense? Okay. So I'm just going to go neutral, 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 neutral. I need like a thing for neutral, but it doesn't. I'm not aware of one. Oh, that's a yep. Yup. Okay. Does that make sense? All right, so if we can make something really unstable by putting negative charges next to each other, and by the way, when we think about stretching it out, which negative charges are interacting? Which of these glutamates are interacting with each other? Let's pick like this one. Who is this near? It's going to be near this, obviously, right? And this, because they're right next to each other, right? But it's also going to be near 1, 2, 3, that 1, 1, 2, 3, 4 away, right? Because it's on the same side of the helix. So we'll have some pushing against each other here, but we'll also have pushing against each other between there and there. Questions? Yes. Yeah, so I think what the question is, if I do a test question, would it be this obvious in terms of the pKs and everything being negative? Yes, for this. Okay, but we're working up to something that's a little more complicated. So this is a helical wheel diagram, and what it shows is kind of where the side chains would be on an alpha helix. So we start here's position 1. There's where the second one is. So it's going to stick out that way. Here it's going to stick out here. Here it's going to stick out here. And what we're doing is we're working our way down in a right-handed manner down this helix. Does that make sense? First amino acid, second amino acid, third amino acid, fourth, fifth, sixth. And so we can see that 5 is 4 away from 1, and they're really close to each other in three-dimensional space, right? If we go like this, that means that 5 is, down below a little ways. So it's maybe about 4 angstroms away. All right, so what I could ask you is I could say, make a very stable alpha helix using the following amino acids. Okay, and what I'm going to do here is these are not connected. You need to connect them in terms of primary sequence. And so let's give you an E and a D and a K and an R and an L, whoops, and an L and a C and I'm going to do this faster. Give you a couple I's and give you a couple L's, and then maybe a B, maybe a couple A's, and then let's do one more. Let's get our tryptophan in there, and we can maybe put in a tyrosine. Okay. So I've given you a bunch of amino acids here. For those of you that are starting to get your codes, what do we know about E? Glutamate. Glutamate is going to be negative, right? D is aspartate. It's going to be negative. K is lysine. Yep, it's going to be positive. And R is arginine. It's going to be positive. And the rest of them are a mixture of, we've got hydrophobic ones, and we've got some more sort of middle ones, and we've got some things that are sort of hydrophilic. I guess most of these are hydrophobic from the way I described them to you before. But don't worry about that. What I'm looking for here is an understanding of what's on the same side of the helix, okay? What's on the same side of the helix, and I don't want you to go to the level where you try to put all the hydrophilics next to each other on the same side or hydrophobics. Just worry about the charges. All right? So you've got a diagram here. Go ahead and try that. How can you get the positives and negatives on sides of the helix such that they all attract each other and help hold the alpha helix in its shape? Okay, so I want you to start thinking about that process. Some people started drawing out amino acid sequences. Some people started drawing sunflowers. But if I was to do this one, and there would be multiple correct answers, right? If I was to do this, I would go, okay, I'm going to start off with, let's just start off with the glutamate because it's there. And now I need a positively charged amino acid 3 or 4 away. Okay, I'm going to go ahead and make it, I guess I'll make it 4. I think I'm going to run out of amino acids. I'll make it 3 because I think I'm going to run out of amino acids if I'm not careful here. But so I'm going to go, so I'll go ahead, I use this, I'm going to go ahead and use the leucine, the cysteine, and the isoleucine. Okay, I guess I'm going to make it 4 away. CI, and now I'll go ahead and I'll put my lysine in. All right, so now I have a positive, and that positive is now 4 amino acids away from the negative, right? So if I was labeling this on my helical wheel diagram, I'd have an E there, I'd have a leucine there, I'd have a cysteine here, I'd have an isoleucine here, and then at 4, I'd have a lysine there. So I have this negatively charged glutamate right next to this positively charged lysine. And they attract each other. If we looked at this in three-dimensional space, if I was to draw a cylinder to represent my alpha helix, E would be here, and let me do it this way. I'll put E here, okay? And I would go around a full turn, and then I'd have K here. And so I'd have, in terms of on the side there, and so then I have this positive next to this negative. Does that make sense? And then I would work my way through the rest of this. So now I'm going to use 2 because I'm worried I'm going to run out. So isoleucine, then leucine, and now what do I need? D, right. I'm going to go ahead, I'm going to put, now I need a negative amino acid to be on the same side, right? So I can get attraction there, and then I'll go, let's go ahead and use 3. Okay, I didn't run out. Leucine, valine, alanine. And I didn't need to use these in the order that they are. It's just easier for me to keep that in my head. And so then now I would do my arginine. And now I've got a positive on the same side, and then I have a alanine, oops, an alanine, a tryptophan, and a tyrosine left. Okay, and this would be my amino end, and this would be my carboxylic acid end, or carboxy end. Questions? Yes, Chloe.
Does it matter, like, is it purposeful that we are putting like positively charged amino acids towards like the carboxylate end of the chain? Is that, would it be like negatively charged?
There is actually a more stable way to do it, and maybe what you're talking about. I don't remember. So there actually is a polarity to alpha helices because of the direction of the of you actually get some small electric field by the direction of the peptide bonds, and we're not going to worry about that.
Okay.
And in fact, I've never really paid much attention to it. After our speaker's talk at the beginning of this a few weeks ago, I feel like I should go back and revisit that because it seems cooler now than it did before. Before it was like, who would care? Who'd ever care? It was kind of over in my head. Now I know why I should care. Questions?
So we could have just put, you could have also flipped the amine end and the carboxyl end and it would still been correct.
Yes, as long as yes, you could have done it whichever way you wanted, and you could do these in that way. If I ask you to draw a sequence and I ask for the amine end and I specify the amine end and the carboxylic acid end, I care, like with atoms. So, but for this exercise, I could flip those as long as I label them. If I ask you to draw a sequence and you draw it backwards, but you clearly but you clearly have them in the right order in terms of the actual direction of the polymer. That makes sense?
Mhm.
It's like for DNA, if you were to draw the DNA upside down to get it all right, it would all be good. All right, well, let's go on and move on to talking about beta sheets. So beta sheets are our second major secondary structure. This is a beta sheet right here. It's an anti-parallel beta sheet. Beta sheets represent the extended conformation of a peptide. All right, so I haven't shown you the structure here, but your hair is mostly alpha helices. If you were to pluck off a hair or just pull out a hair, I can't do it, but if you were to take a hair and you were to stretch it, it does stretch some. Okay, and that's because the alpha helices are actually, they're like springs and they stretch a little bit before they'll break. All right. Beta sheets don't do that. So I really need to own some silk. Silk is actually beta sheets, and so it presumably doesn't stretch. If you own silk, you might have to get back to me on that, something pure silk. But I've never owned any silk. I've always wanted to get some and just never gotten around to it, I guess. But so it's an extended conformation, and it can't stretch anymore because that's all those bonds are going to do for you. Okay. Um, they're flat. They're also held together by hydrogen bonds. So beta strands are held together by hydrogen bonds. And so you can see this here, right? You see how we have the carboxylic or the carbonyl of this amide is a hydrogen bond acceptor from the nitrogen on this amide, right? The nitrogen hydrogen on this amide. And because of the way this one's done, right, you've got this one. Well, you can at least same for any of these, but you've got this. You can imagine you could have a turn here and then go into the next one right away. But what you could also have is you could have this go out here, and you could have a bunch of alpha helix, and then a turn, and some more alpha helix, and then come back here, and then here. And so these amino acids, unlike the hydrogen bonding in the alpha helix, where they're relatively close together, right? 4 or 5 away, or no, sorry, 3 or 4 away. For a beta sheet, you can have things that are very far apart in primary sequence come together when the protein folds to make a beta sheet. Does that make sense? Okay, so these can also be, you can see this says anti-parallel. So in other words, it goes that way, and then it goes this way, and then it goes that way. What a parallel beta sheet would be is you'd have it going that way, and then you'd have it going that way, and then you'd have it going that way. They'd all be going the same way. So you can imagine in a parallel beta sheet, you've got to have a whole, they've got to be far apart. This one goes this way, and then a whole bunch of stuff has to happen. You have to get back over here, so you can go this way again. Some stuff has to happen, so you can go this way again. All right, so they can be parallel or anti-parallel. And if you look at what's going on with these side chains, do you see how they stick up above the sheet and below the sheet? So the side chains alternate sticking above the sheet and below the sheet. And you can see an example of that right here as well. Questions? All right. So here's an example of parallel. So for anti-parallel, we can look at that. And the way we know it's anti-parallel is we can look at the direction of the polymer. And what I do when I'm grading these things, and you can do I draw these arrows, right? You see these are going, if I go carbonyl to amine, I just pick one and do that. And then here, if I go carbonyl to, well, it's not amine, but carbonyl to nitrogen, you see how my arrows are pointing different directions? All right. Whereas down here, if I do carbonyl to nitrogen, they're pointing the same direction. Okay. And something, so one of my favorite questions to ask with this is draw me a parallel or anti-parallel beta sheet. Okay, I ask for one or the other, using 6 amino acids of your choice. Just don't use any one more than once, showing me the hydrogen bonding. Okay,
Can you give us an example of that?
sure, go ahead and do it. Well, we can use the rest of lecture. So right now, draw me an anti-parallel beta sheet using at least 2 strands. Just use 2. So using 2 strands with 4 amino acids in each strand. Do not use any amino acid more than once. Now we only have 4 minutes here, so what you may want to do is try drawing the strands first. So I said anti-parallel, which is the one I've got up so you can look at it. And try drawing the strands first with the hydrogen bonding and then add your side chains. And I'll draw one in the last minute. Then we've got a side chain. Don't worry about lining them up perfectly. It's not an art contest. Side chain along the arm. Yeah. All right, I've got a minute left here, so I'm going to go ahead and try and draw one of these really quick. See if I can do it. Life's not fun without challenges, right? So I'm going to pretend this sheet goes on for a while right here, and then so the important part, I'm going to do the backbone at least. All right, I probably got too many at this point, right? So I'm going to have 1, 2, 3, 4 amino acids that I'm supposed to have. This is where line angle is really useful to use this, because this is the actual conformation. So you can, the up and down thing works really well. I'm going to go ahead and draw the next backbone below it. What did I say, anti-parallel? Okay, so I got to go the other way. I'm not going to actually worry about it. It's not an art contest. So, but I do need my, if this is here, whoops, that's a nitrogen. And probably more than I need, yeah. And I'm going to get all my hydrogens in there. Okay, I haven't drawn my side chains yet. I don't think I made mistakes. I'll check it later. But, so now I need to make some hydrogen bonds, right? I did a horrible job making this easy. I feel like I made a mistake even, but I didn't. I just did a horrible job lining these up. So this one, I can imagine hydrogen bonding to that one. Let's say that, and that. Okay, something like that. So I said it wasn't an art contest, and I'll be forgiving of you. I don't know that I'm super forgiving of myself for messing it up that bad. But if I take this and shift it down, you see how they line up pretty nicely? Right? Okay. You guys have a great day. Let me know if anybody wants to stop by before 4 today.
Was your time at like 3:30? Let me
Yes.
just stop by.
See you then.